So I was asked to first set the stage a little bit. I'm Reisa Sperling. I'm a true partners person, work at both Brigham and Women's Hospital and Massachusetts General Hospital. I've been working in clinical trials as an academic for about 19 years. And I think we're at I'll say an auspicious time in Alzheimer's development. As you know, we've seen a roller coaster of clinical trial results over the past 12 to 18 months. And as a field, unfortunately, as these guys well know, and you in the audience, over the past decade, we've had 11 phase 3 trial failures in Alzheimer's disease. So whoever's doing sports analogies last night or whatever, we're not quite up to the Hall of Fame yet. And the past 6 months in particular, I think, have been a bit of a roller coaster. So the solanezumab study, which we'll talk a bit about, which didn't quite meet its cognitive endpoint in phase 3, and then just within the past month or so, the Merck BACE inhibitor trial. On the positive side, though, I think we heard a lot about the aducanumab and other studies, as well as a number of trials in earlier disease that have started over the past 2 years, and I was thrilled to hear those brought up in all of the previous panels. So all morning and early afternoon, you kept hearing about all the things we should do in the field to accelerate trial development, and we're the panel who's going to say, here's how we do it. So I'm going to ask, we're going to actually start down at this end with Craig. I'm going to ask each of the panelists to briefly introduce themselves, and then they have exactly 60 seconds to tell us if there's one thing they would change to actually improve Alzheimer's drug development. What would it be? So start with Craig.
Okay, so thank you. I'm Craig Ritchie. I'm the Director for the Centre of Dementia Prevention at the University of Edinburgh, and I'm also the academic lead and co-coordinator of the EPAD project. That's the European Prevention of Alzheimer's Dementia project. And I think there's several things that I would change, and most of which we're trying to do within EPAD. But
One thing
that I think is most important in some ways of the EPAD project is the preparation that we have, if you like, pre-randomization into the trial, and that is the development of the readiness cohort. The readiness cohort is going to have several thousand people in it eventually, very deeply phenotyped. It'll provide us not only with patients, which will reduce the screen failure rate substantially, it'll also create probably one of the best resources for disease modeling and clinical modeling with all the biomarkers, clinical data we collect. And thirdly, because of the way we collect the data, we can actually use some of that data for run-in data, pre-randomization, to measure effectiveness again. So the one thing is the readiness cohort.
Excellent. All right.
Hi, I'm Iain Chessell. I'm head of Neuroscience for AstraZeneca in the Early Innovative Medicines and Early Development Organization. My one thing is a bit of a magic wand, actually, but it's all around biomarkers. It's developing biomarker fingerprints for progression in early Alzheimer's disease and really early Alzheimer's disease. And I think, it's a kind of obvious statement. We've all been talking about this today, but this will really help us get the right patients into the right trials and have a meaningful measure of progression, which means we can do more trials. So I think one of the issues we have right now, as I'm sure it'll come up at some point during the panel today, I think we've got a number of targets and we have a number of potential drugs, but we have a real limit on the number of drugs that we can look at in the clinic because the trials are so long and expensive. So we can figure out a way of actually looking at these biomarkers in early Alzheimer's disease and get a readout early on. It'll give us the opportunity to study more drugs. So I'd really wish that to happen. But in terms of how, the EPAD obviously is one example within the DPUK that John Gallagher talked about. At the moment, there's a deep and frequent phenotyping study ongoing to look at biomarker identity in prodromal patients. So we'll get a much better understanding of the phenotype of those patients and hopefully correlate some of those phenotypic changes with genuine biomarkers.
Just under the limit, all right.
I'm Nadeem Sarwar. I'm the founder and president of the Eisai AIM Institute. We make medicines, and our focus is using human genetics to prosecute upon genetically validated drug targets. We behave like a 90-person biotech. We have Zumba on Tuesdays and Pilates on Thursdays. But we're all under one roof, and we bring together data sciences, wet lab biology, chemistry, and translational sciences to prosecute upon human genetic-supported drug targets. So my one thing would be to get better at selecting drug targets most likely to work, and get better at matching those targets to the patients most likely to benefit. And the prism through which I believe we can do that is human biology and data sciences. We can now study humans in unprecedented detail, scale, and speed. And as a result, we no longer have to chase the mouse. The mouse has taken us down paths that were wrong. And now we can study humans. And we can study them in ways we've never been able to do before. But just generating data isn't going to be much use. Applying those data for drug discovery, I believe, is where data sciences are critical. And that's where being able to understand human biology in unprecedented detail and using that knowledge to guide drug discovery, I believe, is the one thing for me.
Excellent.
Like Krams, I had a group of 400 modelers, mathematicians, statisticians, and clinical pharmacologists as Janssen R&D. If there's one thing I could change, I would invite us all to connect the dots. What I mean by this is to take knowledge that we create and integrate it in a way that we learn from each other, borrow information from each other, and the platform to achieve this might enable true prevention in Alzheimer's and true exploration of combination therapies. And that platform is a platform for clinical trial where the patient or the healthy subject eventually becoming a patient is at the center, not an individual compound. And the question is, how do we have to play with what's at hand such that the solution to the real problem, making the healthy person never become a patient, can be achieved?
Excellent. All right, so we have a couple of themes that we talked about when we were on the phone that I am going to explore, and I really am hoping, especially at this point in the afternoon, that we can make this interactive. So please do send in your questions as we go. So the first was, we've heard all day about the idea that we need to move earlier in the disease. And I want to talk a little bit about what the results of the solanezumab and Merck may inform us for this, and in particular, what are the strategies of how we get there efficiently? So I don't know if, Craig, do you want to?
Yeah, sure. So we often hear that the recent Merck and Lilly studies were, critical of the amyloid cascade hypothesis, but I actually think they reinforced them in some ways for those findings. Because if we do believe that the genesis of this disease is 30 years before people get mild dementia, so mild dementia is actually late-stage disease, then it's probably maybe not surprising with what we've learned over the last 5 or 10 years that these trials didn't read out as we'd all hoped. So I think they're reinforcing what we're doing in projects like EPAD and A4 and A5 to go even earlier. But that throws up, of course, key challenges about who are these people and how do we identify them to come into the clinical trials. And I think one of the challenges is also that we've had quite a restricted view of what we call stratification. And I think we have to take a much more comprehensive and indeed complicated or complex look at disease biology in that preclinical prodromal period to be able to then stratify to a much greater degree than we're doing currently to find those patient populations who are most likely to benefit, be on the basis of their genetic signature, their biomarker signature, their cognitive profile. So I think we've learned a lot from those recent, and I don't like to call them failures because they're only a failure if we don't learn anything. They're informative and they're educational about what we should do next. And I think what we should do next is go in earlier, stratify better, and also the third thing is develop much, much more sensitive outcome measures which are relevant to the disease and to the patient.
Right. Iain or others, do you want to weigh in on strategies to help us go earlier more efficiently?
Yeah, so I would completely agree with what you've already said, but I think the focus has been in recent years on amyloid. And the focus has been on amyloid trials and, are we disheartened or are we encouraged by the amyloid trials that we've done recently? I mean, personally, I'm also pretty encouraged because I think there's some very positive signals there. But the challenge of going earlier isn't met by, doing these studies in later populations other than to say we need to go earlier. And I think, we're there now. We've identified the fact that we do need to go earlier. So it is things like EPAD, it is things like the deep and frequent phenotyping, it is things like connecting the dots and putting together all of the information that we're gathering that will give us the information that ultimately we need to generate those selection markers. And I think the selection markers are actually really important. They may be as important as going early because we do talk a lot about amyloid, which is what we've largely focused on today. But when you look at some of the other pathological markers in Alzheimer's disease, and tau being a good example, I mean, we seem to be 5 to 10 years maybe behind the amyloid field with the tau field. I don't really want to be sitting here in 10 years' time saying, oh, we need to go early with tau. We need to find ways of identifying the right biomarkers and the right biomarker fingerprints to get these patients in early. But let's not just focus on amyloid. Let's make sure we spread out and look at all of the markers with the information that we're gathering. And I think, we should have at least a gathering power to be able to do that now with a number of studies that have been run. If we could share all of that data and all of the phenotypic information and all the biomarker information, and we just had a session on big data and actually do some analysis on that, we may actually find that some of the information we need, not all of it, but some of the information we need is already there.
So I agree with that. And again, just to throw some data at this, we do know that half of the amyloid positive normals already have tau. And therefore, I think the idea that we're not going to have to go early with tau is also not correct, unfortunately. I think we're going to have to go early there as well. Do you, Mike or Nadeem, want to say anything about this? So one specific question on this. So it was brought up in several of the sessions that, getting everyone a PET scan or spinal fluid is probably not cost effective and not realistic. But what do you guys think about the way of building algorithms so that we can kind of hierarchically choose people who will get to the point where they're the highest risk and would go on to get an amyloid or PET scan, or tau PET scan or spinal fluid?
So the beauty of thinking about connecting the dot is that platform trials are the last stage of an effort where we think about information that might be available in registries. Then we think about longitudinal natural history studies, and those become instruments to identify a framework of observations, biomarkers, that would enable decision-making in a black and white way. And so for us to achieve the building of these decision-making frameworks, we need to work differently. I'm a stroke neurologist by training, and there's a beautiful quote, history is the point where the imperfection of memory meets the inadequacies of documentation. A lot of people have forgotten that stroke acute neuroprotection is a dead field because it took so long to understand that we needed to go really early, amongst other things. What I would like to see is for the expertise in this room and in conferences to work in a different way, not to hear one talk after another, but to be involved in a week, maybe 2 weeks, maybe even a month of intense designing, where we identify what's the problem we're trying to solve. And I would argue it needs to be prevention of Alzheimer's. And then we figure out what are the different pieces and how do they need to come together to make it happen, as opposed to serendipitous looking, what was it like to feel for the last 10 years? Hopefully we can have another 5 to 10. It needs to be more with a sense of urgency and an engineering mind.
I couldn't agree more. I think simply saying we need to go earlier is too easy, get out of jail free card. Well, sure, going earlier will be better. We know that. And there's maybe 2 interesting learnings we can get from other therapeutic areas. So I see some pretty interesting analogies between where Alzheimer's disease is now and where cardiovascular disease was 10, 12 years ago, and maybe where Alzheimer's disease is now and where oncology was 5, 6 years ago. But those 2 therapeutic areas went in very different directions. So today, a typical phase 3 cardiovascular outcome trial is a 25,000 person, 5-year, mega billion bucks trial. By complete contrast, in oncology, we have potential ends of ones of trials. So those 2 therapeutic areas that have been very successful have taken extremely different strategies. I wonder if we can learn from both and kind of have our cake and eat it. Are there ways for us to be much more sophisticated from a precision medicine perspective, to really drill into why do I believe this sub-patient population is more likely to benefit from this mechanism of action? So just linking that back to something we heard earlier in the day on the session on neuroinflammation. Our bias at the AIM Institute is human genetics. And if you follow human genetics, it leads to immune inflammation mechanisms. It's unlikely that those mechanisms predominate in all Alzheimer's patients. There may be a subset of patients who are especially likely to benefit from an kind of immunodementia therapeutic. I believe we'd be much more sophisticated in matching our precision medicine biomarker to patient subpopulations, rather than try and find the panacea that can solve all Alzheimer's trials.
So you come to a very good point, which is one of the second themes, which is really around how do we speed up the efficiency of these trials. And I want to ask kind of specifically this issue around the large, simple trial, longer trial, and the very intensive, shorter trial. We've seen a lot of companies in Alzheimer's disease bypass phase 2 because phase 2 was not as predictive as we'd hoped, specifically around biomarkers predicting cognitive outcome. So I wonder if people can comment on where they think we should make this trade-off here between large, longer trials or platform shorter trials and what the risk benefits of those two designs are.
Yeah, and I think, sorry.
Well, I'll give a short answer. I know that this came up when we were discussing on the phone, and luckily I can't remember what I said. So I'll probably say something different. So
We have a drug for you.
I think right now it's hard for me to rationalize shorter trials versus longer trials because we don't have the toolkit. We don't have the toolkit to measure progression in a meaningful way in a small population with power. We have we have some of those things, but if you want to do a trial that's going to get you a drug to the market, we don't have the tools. So it becomes a sort of inevitable evil, I guess, that you have to face doing long, delayed start, phase 3 studies, because they're the endpoints that we have in our hands right now. My wish list, having those biomarkers, having that prognostic marker that we can look at to see whether in a trial we're doing something useful pretty early on. And I know most trials are building in exit ramps, building in ways of figuring out futility as early as possible. But even those, they're lots of patients, lots of money, and lots of time. So I'd love for my answer to be, yeah, great, do short trials and do highly intense ones. But I don't think we're there yet.
Well, I know there's a couple people on the panel who may not fully disagree with you, but are trying to push that envelope. So, Craig,
I think what Iain's let's be brave, let's be bold. Let's assume that some of those answers are going to emerge over the next few years. So when we're designing our trials, we're actually incorporating future advances, if that makes any sense. And I think one of the things with EPAD is that there was a discussion very early on in the piece that should we run the cohort study, provide all these answers over the next 5 or 10 years about trajectories, decline, biomarker changes, clinical phenotypes, etc., or should we have our cake and eat it? Should we do the cohort study and in parallel run the clinical trial? And within a singular data environment, you're constantly learning. So I think we have to be bold and be brave and start incorporating, because as Iain said, we may not have the toolkit now, but we're going to get it. And we have to be. The other way to shorten trials, of course, and it refers back to my original statement, was that the trial itself has got there's, I think somebody earlier said, there's as much time spent screening people into a clinical trial as it is following them up. So with a readiness cohort approach, where you're deeply phenotyping people who have consented to consent to being in a trial, one would hope your screen failure rate drops from 90% to maybe 9%, and you accelerate through into the actual randomization phase much quicker. So that'd be another way to nibble a year or two off the duration of your clinical study.
So I think these trial-ready cohorts very much agree with this, and efforts internationally to do this. Of course, there are some tricky things about how you keep these individuals in a trial-ready cohort when they all really are joining because they want to be in a trial tomorrow. So, but I think building the infrastructure really makes quite a bit of sense. Now, we talked a little bit about the biomarkers, and so I really want to get to the nitty-gritty of that because, of course, there's biomarkers for selection, but it sounds like what we're all asking for are what I'll call theragnostic biomarkers, you may have a better term, but that actually predict cognitive outcomes. And that's where I feel like we have sadly not, we're not quite ready. So we've seen dissociation between biomarkers and cognition in both directions in recent trials. So.
Can I perhaps come back to the previous point and connect it to your question?
Perfect, connecting the dots.
Gosh, and so the idea that a trial should either be small or big, that's the wrong way of dichotomizing. The way I look at it is, I have a research question, what is the most appropriate way to answer the research question and base a decision on it, such that they make the correct decision at the earliest time point in the most efficient manner. And if I do my work, I'll get some experimental design, and that's the right design. Now to the biomarkers, the instruction that we provide to our translational medicine groups is, can you please help us? We are designers for clinical trials. And you know what? A small trial with an endpoint, clinical empirical endpoint that isn't fit for purpose to start with will not make for a good proof of concept trial. In other words, what we need from you is some biomarker observation that will help me to have an early observation, and it can guide the direction. So I'm making this up now. Say if you work on a BACE inhibitor and you want to know initially, is the molecule that you're working with able to reduce A-beta-42 in spinal fluid? You don't need 10 years, you need hours. And if you then start to connect the dots, you know, this one observation with another observation, with another observation, you end up with a framework that is not one simple observation, but it's a framework that ultimately helps to make the correct decision at the earliest time point in the most efficient manner. And perhaps one way of going about this is to think more about seamless processes, where we don't have one siloed phase 1 study, one siloed phase 2 study, one siloed phase 3 study, but a universe of research questions that we are ticking off in an integrated manner.
That's great. All right, let me, I'm going to bring up two questions that came from the audience that I think are relevant to this. So one is, are there any lessons learned from the recent Parkinson's trials that might be relevant for AD in this? So those of you who might cross paths, do you want to take that one?
So actually, joining the dots to what you just said, I think there's two opportunities. One is around being more sophisticated in what we call a biomarker. So some of the themes we've heard today, you know, can we get to a paradigm where our primary biomarker isn't the really difficult imaging? There's a sequential path that you can go through where things can be measured at home, in the waiting room, by blood, by CSF, and then by imaging. And hopefully that triage gets you, instead of 90% failure, to a much smaller percentage of failure. The other way is to better mitigate for risk. So if we really believed, if we were 100% confident this drug would work, but we had to do the 20,000 person trial to do it, we'd do it because we'd be really confident it would work. So how can we manage that risk? And one example in PD that I believe is interesting is, and it was mentioned earlier, is GBA, which is a very interesting genetically validated drug target, potentially for Parkinson's. But more immediately, you can test it in a rare disease setting. You know if it doesn't work in that rare disease setting, it definitely won't work in Parkinson's. So you can take a stepwise kind of risk mitigating model. So in Alzheimer's, are there ways we can be more sophisticated about testing our hypotheses, not necessarily in the kind of moonshot Alzheimer's population, but in an incremental way to mitigate our risk?
All right, so the second question, which I hope will help us out of our scone-induced postprandial coma here, is actually a controversial one, but I think it's relevant. So you've heard a lot today about this go earlier, but the question from the audience is, if the goal is to go earlier and prevent Alzheimer's disease, should we simply forsake research and treatment benefits for patients today, symptomatic patients, and is that ethical?
Absolutely not. Because if you look at it, we start with a belief system. Initially, our belief system was we can take certain types of compounds and test it in full-blown Alzheimer's dementia. We've shifted in one direction, we're continuing to shift. We shouldn't be just as dogmatic as we were some years ago in one direction, in the other. The real question is, what is the correct intervention timing for a particular treatment, depending on the mechanism that we are trying to modulate, to get the desired result? And so there is no black and white, early or late. It should be a research question, and we should build research frameworks to understand what is the latest time point at which we can intervene and still be successful.
So that leads into one of these other themes, which is combination therapies, because this is my own bias, but I think one of the ways we don't forsake patients who are already suffering from this disease, who not only have amyloid, not only have tau, not only have neuroinflammation, but neuronal loss, how do we get to combination therapy for multiple mechanisms?
So I think we have a simplistic sort of view of a starting point, if you like, for the disease processes that lead to dementia in later life, and the Clifford Jack slides are obviously well referenced in that regard. And I think what we have to do is to put empirical data onto those curves to see whether or not there is this sequence, or whether or not there's the co-occurrence or the co-location of disease processes at the same point. And in doing that, we might find through that sort of systems biology approach, a particular node which is relevant to drug, which will affect the cascade thereafter. It's highly likely, however, that any individual's clinical phenotype is driven by one or two, or at least maybe even three disease processes. So switching off one of those processes with a very specific BACE inhibitor or monoclonal antibody will probably do very little for the clinical phenotype. So I think we're absolutely committed to seeing mechanisms or ways we can actually do combination therapies. I think to do otherwise would be, for what is an incredibly complex chronic disorder, monotherapy as a silver bullet. I just don't think makes any logical sense.
I think we have to face that it's going to be essential. Every patient will have a slightly different presentation with slightly different underlying molecular biology, one would anticipate. I guess one thing I've thought about is, can we learn anything from oncology? Where increasingly, in immuno-oncology, we're mixing different agents together in trial populations. And I'd love to say, I thought this through enough to say this is what we can learn, but I think the difference there is you've got a measurable endpoint that you can literally measure. You take a ruler and you can measure it, and it's either getting smaller or it's not. And I think that again brings us back to the need to have an endpoint. And in this case, for combinations, maybe it isn't a prognostic endpoint. Maybe it isn't something that correlates perfectly with cognition, but maybe it is something which is meaningful enough to be able to say, this base and this monoclonal antibody, they work better than this base and a different dose of this monoclonal antibody. And there we can start to build a data set. I think, the danger is always to be sucked into, we'll do all this preclinically. And you can see some fantastic observations preclinically, but I think maybe I'm doing the preclinical models a disservice, but it's hard to see at the moment how the state of the art with preclinical models can recapitulate some of the aspects of the disease that we're seeing in the clinic. So I think we have to build these data sets in the clinic, but we have to be smart about doing it. And as the biomarker field does develop, and we're never going to stop, we just have to put our foot on the gas as we start to pick out things which are tantalizing, but maybe not the magic bullet we need. Maybe they're not the one the FDA is going to say, if you have an effect on that, we'll give you a label. But if it's some interim, then we can start building combination studies in and start to get some data on some of those biomarkers to give us an indication of where to go.
Right,
I would hope.
The accelerated approval mechanism.
Yeah.
So we have talked a little bit about endpoints, and we heard a lot today about iPhones or other clinical endpoints that are continuous and passive. And then we also heard about the idea of time and time to event, which I think is biased here, but problematic in Alzheimer's disease, because you just don't go to sleep with preclinical Alzheimer's disease and wake up with MCI, or next weekend you move from MCI to dementia. So I wonder if people can talk a little bit about how we get to more efficient cognitive clinical endpoints, realizing that this is a continuum. And if you have thoughts, this is a not prepared question, so making you guys think on the fly here.
One important element in this is to be clever on how we analyze the data. If you look at the Diane population, if you were to just look in an absolute way at time trajectories of individuals, that would be one way of making sense of the data. But if you then condition your analysis with other available biological information that is available for each of the individuals, all of a sudden you realize, hey, here is one family of trajectories and here's another. In other words, use modeling techniques to integrate information and make sense of it. And we heard it about the multiple sclerosis world, how important it is to identify subsets of patients. We will have to learn to do the same and move away from more empirical pairwise comparisons that are quite simplistic.
So modeling designs. Other thoughts on how we do better on the saying this is clinically meaningful? So one question is also, as was brought up, regulators fortunately have really moved to allowing cognitive endpoints in these earlier studies as the primary outcomes. But then we are left with this issue of how do we determine clinical meaningfulness and what are the best methods or new methods that we might need on that?
Sorry.
Go ahead.
Well, I think the clinical meaningfulness is a sort of a secondary question to getting the right cognitive measure in the first instance. And I think one of the challenges we've had with preclinical prodromal studies is we've tended to take the rating scales we've used for dementia and try to bring them back and fit them into a preclinical prodromal trial. And I think, come back to your point about toolkits, what we needed to do is actually develop a new toolkit that worked in the biology forward to the cognitive measure, rather than what we had available to us backwards. And that's certainly what we did in EPAD. We spent 2 years developing the EPAD cognitive evaluation from a biological perspective with experimental neuropsychologists, rather than just trying to fit in what was out there already. Clinical meaningfulness is really tough in preclinical because by definition they're preclinical. So we have to compel or convince that, reducing the progression of cognitive impairment is in itself a worthy outcome.
So just looping back to from the first session, I think it was Vivek who challenged us to think about alternative measures of success in a clinical trial. And we often use this phrase, unmet patient need, in kind of clinical application. And maybe just taking that to the extreme, who should decide that? At the moment, we try and use endpoints that a regulator agrees that's the right endpoint, or a payer says, I'll pay for that, or a doctor says, I can measure that. Why don't we ask patients what they want, and then try and make drugs for what they want? So I was very struck earlier today when you had the patient on stage, and there were clear traits and symptoms that I feel would have improved that patient's life, the ability to smell better, the ability to sleep better, etc. So I wonder whether it's a bit too simplistic to think about a single Alzheimer's endpoint when it's such a complicated disease. And can we instead think about what are specific things that would make patients' lives better as defined by the patient? And how can we better react to what we often use this phrase of unmet patient need?
So I love that, partly because one of the few advantages, it's a lot harder to go earlier and earlier in the disease for all the reasons we've said, but one of the advantages, people can tell you themselves what they want and how they're changing, and have that be what's meaningful to them. Sorry, go ahead.
So what I want, for one, is to be as brilliant as I was when I was 18 years old, when I'm going to be 81. And that's easy, but it'll take 80 years or 60 years to look at. And so the problem that we have here is the frog that jumps when it's hot and gets boiled to death when the temperature rises slowly. So what we need to figure out is how to convince people that it's okay not to smoke, to take antihypertensive medication, and you know what you get? You don't die of a stroke or a myocardial infarction, but I can't show you in the next 6 months.
No, you die of Alzheimer's disease. So we've got to fix
Bad
that.
for me. Yes. But so in a way to shift that framework from what's obvious to anybody to some very early observation that still seems valuable. How do we connect that?
So actually relevant to how do you get to, at 81, the same memory as 18, one of the questions from the audience is neuroregeneration, the answer. And what would a trial look like designed for that? A lot of smiles and nodding.
That's a tough one.
Absolutely. And I think we don't know, we haven't been able to stop the pathology to the extent that we can really gauge the plasticity of the brain to regenerate in the absence of the toxicity. We may have indications of that from the aducanumab study that the A-beta's production has been reduced with the BACE inhibitors as well. But if we don't look for it, if we don't look for the increase in synaptophysin and the increased synaptic connectivity properly, we won't measure it. We're going to start doing some of those things in DFP to at least have the biomarkers to measure those things. But I think we've got to turn off the pathology first before we worry about regeneration. Let's hope we can.
So one other thing that we haven't gotten to, but I think is important, because we've all been talking about our ideal trial designs, but these do come with dollars attached. So doing especially big trials that involve all of these biomarkers, how do we change the economics? And we heard, some pretty daunting figures of investments of $300 billion from certain companies. So just thoughts on how we can actually change the economics so that none of you give up on Alzheimer's disease before we get that successful treatment.
So it comes back to connecting the dots. We all have good intentions. But if we look at payers, investors, patients, etc., they're so disconnected in their individual utilities that it's difficult to have a sensible discussion that says the economy is going down if we don't get together and work together, if we don't stop thinking in silos. So I think the most important piece is to get the relevant people together, compare utilities, and integrate them into one guiding utility. That's currently not happening. Payers have one interest, regulators another, etc.
Yes.
I think another way for me, again, not wishing to sound like a broken record, but right now, our choices are limited. right now trials are big and expensive and long. For me, speaking personally, we have a limited number of goes because we have a limited budget. And when we've spent that budget, we don't have any more goes. So we've got to be really careful about which one we choose to have a go with. And I'd love the situation to be where we can have 10 goes instead of 1. But the way of mitigating that is to do things, connecting dots in another way, do things together. Partner assets, have peer pharma collaborations, academic pharma collaborations, but actually be a lot more collective in the way that we address these things, not only in the studies we do, but in sharing the data that we get out afterwards. So actually everyone can get a common understanding of what's been achieved. I think that's happening. It's great to see a lot of data being shared now. But in the short term, that's a sort of quick fix. In the long term, as trials do turn in from these very long trials into shorter ones, we will get more shots on goal. We'll get more chance to test the targets that we've talked about. And we'll have more chance of actually winning.
So there's one question which is, maybe we can have a short answer, but it's about, is there a better word or terminology than prodromal to trigger early clinical intervention? And in fact, I just had this question on terminology because you use preclinical as meaning mice, which at least in the United States, that's non-clinical. Right. And we use preclinical as preclinical symptomatology. But I think it does really bring up this important issue that the terminology is confusing. And I can say there's a number of groups, the NIA, Alzheimer's Association, the International Working Group, all trying to figure out how you name a continuum that may occur over 2 or 3 decades so that we all know what we're talking about.
And I think you've just said what I was going to say. It's really hard that we've taken a continuum of disease and we've tried to put it into buckets of preclinical, prodromal, dementia, mild, moderate, severe. And we've also done the same thing with our biomarkers, saying amyloid positive and negative. Doctors love things that are binary and in groups, but it makes no sense from a biological perspective. We should look at the continuum of amyloid disease, we should look at the continuum of tau disease, we should look at the continuum of clinical features. And getting away from these stages may actually help us move forward.
I do think the definitions are really important because I find it confusing. I mean, everyone refers to different stages of Alzheimer's disease as in different ways. I mean, my other love, this sounds a bit weird, but my other love is pain. So I spend a lot of time looking at pain control. And actually one thing that the International Association for the Study of Pain did was on their website lay out definitions of, what does neuropathic pain mean? What does inflammatory pain mean? We need to do that. We need to actually lay out a set of definitions so that everyone says it's talking the same language. Right. It sounds like a minor point, but actually, even in a room of a small number of experts, prodromal, early, preclinical, are we all talking about the same thing? Are we all talking about something different? So getting those definitions right, I think is really important.
Right. My early AD is 20 years before many people's early AD.
Exactly, yeah.
So there's a number of other questions, but we're down to the 2.5-minute mark. And although we've talked about the challenges, I thought we might end by giving each person 30 seconds, even half the time you had before, to say what's the one thing that makes you most hopeful about, in this time of uncertainty, about AD clinical development, so that we reconvene in 5 to 10 years from now, that we're not having the same debate. So I'll start with you.
When Husseini Manji and Roy Twyman interviewed me 7 years ago, I said, I have this crazy idea. It's about combination therapy in Alzheimer's. I want to prevent Alzheimer's. I want to do platform trials. And Husseini said, you know what, calm down a little bit. We'll get it done. And here I sit next to Craig Ritchie, who leads IMED. Diane is happening. I'm incredibly hopeful that we're going to solve things. And I can only say, with this type of expertise in the room, let's get together in a different way and start cooperating in a more intense, intentional, designed manner.
Nadeem?
A new culture. Dementia doesn't discriminate. It doesn't care where you're born, where you live, where you're from. And this room reflects a new culture to try and defeat dementia as a collective. The only hope we have of doing that is if we break boundaries and if we innovate without borders. And this room signifies that across sector, internationally, across technologies. So my real hope stems from a really new culture for tackling disease.
Awesome.
So for me, I think we're getting there. The recent trials, I'm optimistic about, not pessimistic. I think we've seen, from aducanumab, from solanezumab, we've seen hints of signals that say we're doing the right thing. And we all accept that the populations that those studies were done in have 20 plus years of accumulation of amyloid. It's incredible to me that actually we've seen anything. And the fact that we have gives me huge hope. I mean, I think that's a real turnaround moment.
And I think everything we've just said there, we're talking to each other, we're having the right conversations, and dare I say, we're asking the right questions now. We might not have all the answers, but I think in the room and outside of this room, people are asking the difficult questions and putting in place the mechanisms to answer them. And I think projects like Diane or the A4 programs, if you talk about EPAD, you talk about DPUK, they're fantastic public-private partnerships as well. And I don't think we should be ashamed of recognizing the incredible value of the academics and the public sector and the funders and the industry people coming together. I think that's what makes me feel very optimistic about the future.
And we're within 2 seconds of the red flashing light. So I'll end there and thank all of you from your excellent ideas.